Hello, this is Eva Amson welcoming you to this Bite-Size Bio webinar, which today is sponsored by Leica Microsystems. Today's presentation is titled Hyperplex Cancer Tissue Analysis at Single Cell Level with CellDive, and it's being presented by Dr. Nicolas Mouchet of the University of Rennes and Dr. David Pointu of Leica Microsystems. Nicolas has been co-manager of the H2P2 histopathology platform at the University of Rennes since 2019. He completed his engineering degree in 2006, gaining experience in molecular genetics and RNA expression analysis. During his PhD and postdoctoral studies, he trained in cancer research bioinformatic analysis. In 2017, he earned a master's degree in computer science to better support the evolving cross-disciplinary nature of scientific studies. David has been application manager for CellDive and translational research at Leica Microsystems since 2021 and has broad experience in light microscopy, including various aspects of cell and tissue imaging and analysis. After studying physical chemistry at the University of Strasbourg, he completed his PhD in 2002, followed by a postdoctoral fellowship at the Institut Curie. In 2004, he moved into industry and has worked as an application specialist in high-end microscopy at several companies. As always, we will have a question and answer session after the presentation today. So please type any questions you have into the questions box, which appears on the bottom of your screen, and I'll put them to Nicolas and David at the end. So now over to you, David, for the presentation.
So thank you. Good afternoon, everyone. It's a pleasure to present you and to introduce you the CellDive technology. So you will see in this presentation, the CellDive system is an Hyperplex imaging solution that will give you the ability to multiplex and visualize the contextual relationship between the key proteins in the same cells from a single piece of tissue that will really help you answer a question that you probably did not have the tools for. We can now investigate the complex protein networks without disrupting their cellular relationship to the surrounding tissue. And for that, we are using a proprietary methodology that I will describe to you to assess the multiple proteins in a tissue section at a single cell level while preserving the tissue context. So the plan for my presentation today, I will introduce the goals and aims of the hyperplexing. I will explain the workflow. We will see how we can define the study. I will touch about the advanced calibration and the image processing, which is also fundamental in this kind of technology. I will then introduce the ClickWell, which is a new slide holder with a cover slip-free device. And then I will show you the imaging workflow to make the process more concrete to you. So the goal of hyperplexing, if we start where we are for now, most of the things were done in terms of analysis to answer, is it cancer or not, as you can see on the H&E image on the right. The power of hyperplexing, actually, if we overlay the same image with the hyperplex fluorescent experiment, will help actually to answer what cell types are present, where are the biomarkers expressed, and what features define the tumor microenvironment. Is it vascularized? What's the immune cell infiltration in the tumor? What immune cell types are in, out, or around the tumor? And then that will help the researcher to see and to characterize the cancer, the tumor microenvironment that can guide through the best therapy to use for a specific cancer and also for a specific patient. It will allow to explore the spatial relationship. In terms of the workflow, so the CellDive technology, it's not only an imager, it's composed of different things assembled together. So we will start with the antibodies. It's a big part of the CellDive technology because we spent time and effort to validate a lot of different antibodies. So that means that the antibody that we validated works. They will bind the expected epitopes. And validation means that we validate the specificity of the antibodies the sensitivity and the detection through the microscopy system, and obviously the compatibility with the cell dive process. There is no need of reagent kits or predefined antibodies panels. They are simply not required. You can still work with the antibodies that you are used to work with. And we also develop a specific antigen retrieval process. Then we enter the cell dive process itself, which is an iterative imaging process where the chemical dye inactivation process will occur, so that will quench the dye molecules and enables the sample to be probed repeatedly with new dye conjugated antibodies. This method will preserve the integrity of the tissue over the multiple states. Then we have the cell dive imager that you can see on this is the red box with a specific acquisition software that I will describe a little bit later. And after that, we'll be ready to start the analysis. And for that, we partnership with Leica Lab, and we offer the Halo software to perform the in-depth analysis of the tissue that you will study. More details about the workflow. So we can work with a different kind of tissue type. They are all FFPE tissue block. We can work with the whole tissue. We can acquire and image the entire tissue on the slide. We can work with the different regions of interest that will be defined by the user. And we can also work with the tissue microarray, which is also a good way to scale up this kind of technology. Then we will enter the iterative process. So we will always start with an autofluorescence imaging. Characterization of the autofluorescence is really fundamental because depending of the tissue type, the autofluorescence signal can be pretty high, and it can compromise the signal of a given biomarker. So it's very important to characterize that. Then we will perform the staining, so with different conjugated antibodies. Then we will use the imager to image the biomarker that we just stained. We are using a 5-channel instrument. That means that we will be able to acquire a DAPI signal plus 4 biomarkers for every cycle. Then a lot of image processing and alignment will occur in the background with absolutely no user intervention. After that, we will perform the dye inactivation. So it will inactivate the fluorescent dye, that the antibodies will remain on the tissue. And then we will be ready to start a new cycle after a new characterization of the autofluorescence. Once we did the expected panel that you define, we'll go to Halo software platform to perform the image segmentation and the analysis, doing the classification, doing the segmentation and the clustering, and at the end, generated the data. In terms of biomarkers, the validated biomarkers are from different targets. So you will be able to choose the number of biomarkers and which biomarkers. And then we have validated different biomarkers for human and mouse tissues. They can be either monoclonal or polyclonal. There are markers for tissue architecture, cell types, cellular structures, also segmentation markers. Of course, a lot of biomarkers for a variety of cancer types, and also biomarkers of drug resistance, immune response, and angiogenesis. So you will be able to pick up the antibodies that you need for your study and to carry out the study with the cell dive system. Then in terms of imaging, we have developed a specific advanced calibration and image processing with absolutely no user intervention. And all those corrections that you can see on the left will help actually to correct the raw images that I acquired with a microscope. Those images will become really artifact-free. They will be stitched on the fly during the acquisition process. And you will be able also to get the different cycle all together thanks to an automatic registration and alignment that will be performed by the acquisition software. You can see here an example of the accuracy of the process. You can see, for example, in gray and red, those are DAPI and pan-signatory-keratin acquired during round 3. And in blue, this is the Ki67 acquired in 1-1. And you can see the high level of accuracy and precision during the alignment and the registration of the different cycles. It will allow you to get such a beautiful image. So here it's a colon cancer tissue with, in total, that there were 33 biomarkers. Here in that picture, there is 11. And most of the biomarkers are to study here the cancer immunity and progression within the tumor. In terms of slide order, we just introduced a new slide order called the ClickWell. That's the elimination of cover slipping and decover slipping that will reduce the cycle time, and it will help to preserve the tissue integrity. This slide order will allow you to work and to keep the slide always in this order and to perform the different labeling, washing, or dye inactivation. But you can also remove the slide and use automatic stainer that will accept the slide. And after the process, you will be able to put back the slide, and the accuracy has been made to be recognized by the software and to continue the iterative process. So it will really open the flexibility and will fit well what you have in the lab to perform this kind of staining. In terms of the process, now I will show you a video where I will use the software, and that will give you a better idea how the cell dive imager works. So let me start here. So here you can see the full process. So after you prepare the slide, you will design the protocol and add the slide and create the batch in the acquisition software. We'll start to make the protocol. So here you will create a new protocol. You will give a specific name. Then you will tell the system if you will image through the slide or through the cover slip. So now we are imaging more and more through the slide. You can then define the antibodies that we will use, and with the current spending, wavelengths, and fluorescent dye. Here you will pick up the name of the different antibodies, and thanks to that you will keep a track of precisely of the experiment. And then you will also adjust, if needed, the exposure time. You can also enable the creation of a virtual H&E image that can be beneficial because it will help the pathologist to recognize the region of interest before to start the multiplexing experiment. And then you will add as many runs as your study will need. Then you will publish the protocol, and you will select the slide that will be processed using this protocol. So a batch is created. And then we'll be able to start the process. So the software keeps a track of every different project and slide where we are. So for example, with the current slide, we are doing the sample preparation. Once it's complete, you will tell the software, then you will put the slide into the imager, and we will start the acquisition session that will start with a 2x whole slide imaging, a fast acquisition in order to see where is the tissue and to define the region of interest to be acquired for the further rounds. So here you put the slide. We will read the barcodes of the slide order to recognize which order are you using. And then the system will recognize the slide. Every slide are barcoded. So one slide has been detected here, and we will start the 2x acquisition. So the preview is being acquired. So here you can see the tissue section. You can zoom in, zoom out, change the brightness, and then you will define the region of interest that you want to acquire. So it can be done directly from the 2x, or as an option, you can also use a 10x if you need a better resolution, a better view to select with accuracy the region of interest to be acquired. So here we selected different fields. We save it. And now, because we selected the option, we will use the 10x. So the 10x will image again the area that has been defined with the 2x. And then we will use this to select the final area to be acquired. So we put again the slide into the instrument. It will recognize the slide holder and the slide itself. It's running a tilt check. It's to check actually if the tilt is in the boundaries that we define. And now we started to scan with the different wavelengths. Here we are just using 2 wavelengths. So we accelerate the acquisition. So we are still using the 10x. And then we'll be able to select the region of interest to be acquired using the final magnification, which is 20x. So the scan is complete. And then the viewer will be open. So first we will, after each imaging session, there is a quality check control made by the user. So you will open the viewer. So here it's to double check the acquisition went well and to see if you are happy with the focus and the stitching. So you will explore the tissue to double check that. And then if you are happy with that, you will press pass on the software, and then we'll go to the next step. So next step would be actually using again the images that has been acquired with the 10x. But thanks to the 10x resolution, you will be more precise to define the region of interest. So here you will drag and drop as many regions as you want, as you need for your study. And then that will be the final, the area that will be used with the 20x. Just accelerate this. So now we'll start the iterative process. So that we'll start to do the autofluorescence characterization with the 20x and with the fluorescent channel that has been selected depending of the different antibodies. So again, we read the barcode. Again, we check the tilt of the slide. Then we run an auto-alignment. So the auto-alignment means that we map the coordinates that has been defined with the 10x to the 20x, and now we'll be ready to acquire the entire regions that we selected. So now the full scan will occur. So here we are using the five different fluorescent channels. And again, speed up the video. Then you can see in the software where we are, because the software allows to use different or multiple slides and multiple projects. So now we are double checking the image quality, the focus, and the stitching after the autofluorescence characterization. So yeah, here you can use the viewer, you can overlay different channels to double check the structure and to recognize the cells of interest to be sure that everything went well. And then we'll be ready to make the biomarker labeling. Once it's ready, you will put again the slide into the imager, and then we'll start the biomarker imaging using the 20x. And thanks to the fact that we acquired the autofluorescence signal, in the fly, the autofluorescence will be subtracted from the biomarker images. So the user will get the final images with the autofluorescence being removed. And we will do that for at each cycle. That means that at the end of the latest cycle, you will get all the final images corrected, stitched, aligned, and ready to go to the analysis software and to use the Hello platform. And of course, after this acquisition, you will have a quality check control after the 20x biomarker imaging. And again, you will double check focus, stitching, and also if the labeling was a success. And if it's successful, you will select pass, and then you will be able to start the next round. So it's time to conclude about my CellDive presentation. So we have seen that the system is customizable. That means that the combination of the validated antibody list and the CellDive process will enable you to customize your multiplexing experiment to fit your research question. You can just pick the markers you want and pick how many markers you want for your study. And the CellDive solution is easily scalable. You can run a single slide, or you can interleave many slides using either a manual process or an auto stainer for the slide staining. CellDive is also precise, thanks to the microscopy method that we have developed with the advanced calibration, the image correction combined with our autofocus method. It will ensure that all your multiplexing data is seamlessly stitched and precisely aligned during the acquisition, enabling downstream single cell analysis. We have also seen that the biomarker expression is also very precise, thanks to the characterization that we carry out about the autofluorescence, and this is fundamental. So then when using the CellDive system, you can very precisely determine what areas of your tissue to image. If the whole tissue imaging is important, you can simply acquire the entire tissue, or you will be able to acquire just the areas that you are interested in. The CellDive system is proven. It has been rooted in for 10 years of development and scientific collaboration with a significant number of peer-reviewed publications, users in different countries, and a comprehensive list of compatible antibodies. And we partnered with Indica Labs to offer Hello as our analysis solution. Thank you very much. I will let Nicolas take the stage now, but before I would like to thank him as well as all the people from the H2P2 platform. Thank you very much.
Okay, thank you, David. I'm Nicolas Mouchet. I'm the platform manager from the H2P2 platform in bioscience at the University of Rennes, and I will talk to you about the Hyperplex cancer tissue analysis at the single level with the CellDive. And I would also like to thank Leica to give me the opportunity to talk about this topic. First of all, so we are a core facility, an histopathology core facility with a lot of sponsors, as you can see on this slide. And I won't name them, but they will recognize them. Where are we? So we're near Paris. We are at about 1.5 hours by train from Paris. And more precisely, we are in Rennes at the medical school. And what do we do? Well, in fact, you can come to us if you have samples and you want to discover whatever you want to look at. So you bring your samples, and then what we can do is the dissection and the embedding. So we've got fresh or fixed sample. Here, for example, we got a rat brain. And then from this rat brain, what we can do is to do a purified embedded sample or a frozen sample. From the sample, then we will do a sectioning with a microtome at room temperature or with a cryostat at -20. And then once sectioned, then you put it on a slide, a glass slide. Once you've got your sample on a glass slide, then you will want to do a staining with a routine stainer. We've got one from Leica, or you can do an automated IHC on immunofluorescence. And then your sample is stained on the glass slide. And then you can do the acquisition. And for that, we've got two scanners, one from Hamamatsu and one from PG Stage, which this one is a confocal scanner. And once you got your image, what you want to do? Most of the time, you want to do an image analysis and measurements. For this, we got many software. The most of them are Hello and Amira, but we also have open source software as QPath, PG. And if you have any questions, then you can answer them to the image analyst. On the facility, we also have technological specificities. For example, here you can find that we've got the tissue microarray. So you've got many blocks of paraffin embedded tissue, and then you pick up a small part of the tissue and you put it on another block of paraffin. And then you can section it, and then you can have up to 300 cases on the same slide. We can also do laser capture microdissection. For that, you bring your fresh frozen sample, and then you can do a small microdissection, and then you capture a cell type. And after that, you can do RNA, DNA, or protein extraction. We also have this specificity that we can do tissue clearing. Here, for example, you've got a kidney or brain, and we do sectioning, but we can do on the whole organ. And once it's sectioned, or if you got the whole organ, then you can do the clearing. And here, for example, just on the right, you've got an example of the imaging after the clearing. And the last specificity where I will show you is the Raman spectroscopy. This is a non-invasive way to study your sample, and then you can look at the differentiation, the diagnostic, prognostic, and you can decipher which kind of bacteria you've got, for example. And what's new since 2021? On the core facility, in the partnership of Leica, we've got a new device, the cell dive imager. And why the cell dive? In fact, with the cell dive, you got the ability to map the sample on the whole slide in depth answer of research questions. You also have the ability to make up to 60 markers and even more. And you've got a list of tested antibodies that is available and can be completed with our or your favorite antibodies. And the technology, as told you, David, just before, has been proven for many years and developed and constantly improving. And we are connected with the research and development team and the field team from Leica. On the platform, we've got many projects. This is for them, and some are looking for the study of the tumor microenvironment in follicular lymphoma, others with the identification of low-grade human hepatocellular carcinoma, inflammatory microenvironment of hepatocellular carcinomas, and the characterization of human subtype in TRP-negative breast cancer. I will only show you one, is the study of the tumor microenvironment in follicular lymphoma, and a small part of the study. This is a tonsil tissue. Here we've got 9 colors that are off of the Leica list, plus the DAPI. Our objective was to reach 15 stains, but with the 9 colors, it's about 3 days of work, included slide preparation. For this tonsil tissue, I told you we've got 9 colors, and this is how they are represented on the Allo software with all the stain and the false color we gave them. I will show you 2 groups of markers, and I will go in details just after. For the first group of marker, I will begin with the CCL2. That is a member of the small induced gene family, and CCL2 plays a role in the recruitment of monocytes to sites of injury and defection. The next one is CD 163, that is exclusively expressed in monocytes and macrophage. CD 68 is 110 chylodietin transmembrane glycoprotein that is highly expressed by human monocytes and tissue macrophages. CSF1R, that is expressed in mononuclear phagocytes in the brain, known as microglia, which plays a role in neural and brain development, as well as in the osteoclast, which plays an important role in bone mineralization. Phosphorylation of CSF1R triggered by ligand binding to the receptor activate multiple downstream kinase pathway, including PI3K, ORF1 and 2, and GINK. And if we do a merge of those 4 markers, then we can obtain this image. And if we add DAPI, then we got this image. Then we can study it, as I will show you just after. The second group of the 4 markers. So we start with the CD8A, that is selectively expressed in a subset of T cells, and in a structure is a multimeric macromolecule composed of individual disulfide-bonded subunits. CD8 alpha is expressed by most T lymphocytes with cytotoxic or suppressor function. CCL21 is a ligand for CCR7 and guides the interaction between CCR7 plus T cells and antigen-presenting cells, APCs, needed for T cell education and priming. These events are involved both in triggering adaptive immunity and maintaining peripheral tolerance. CD20, as known as MS4A1, is a human B lymphocyte surface molecule that is widely expressed during B cell ontogeny from early pre-B cell developmental stages until final differentiation into plasma cells. Core4, also known as CMA4D, is a member of the semaphorin family and function in axon-guidance, immune response, cell migration, cell-cell communication, tissue development, and angiogenesis. As you can see, we've most focused on the immune response, and this is the merge of the second group of 4 markers, plus now the DAPI. And once we merge all of them, we can obtain this kind of image. And what we'll do next? Then we use the Allo software. And I will show you only one possibility of this wonderful software. This is the nearest neighbor of two markers. So from one image, we can draw an annotation node zone where there is specificity. And once the annotation is done, then you can do the segmentation. And from this segmentation, then we can study what I just told you, the neighbor study. For here example, we got all the plots of the CCL2 from this annotation zone. And I've added the CD163 plots from this zone. And then if you got the merge of it, then you can see all those plots are in the annotation zone. What we can do next is to see the neighbor, the nearest neighbor. And this is the line that you can see between a CCL2 protein and a CD163 protein. The lines are starting from the CD163 to the CCL2. And all data are possibly saved on a spreadsheet for further analysis. Then we also have the possibility to see the co-localization of those two markers. And if you remove the plot, just let the line, then we obtain this image that is showing all the repartitions of the nearest neighbors. And if you had all the stains on this image, then you can see all the repartition of the stains. And this is, I think, a beautiful image. But now it's up to the researcher to decipher those data. Next, I will show you just one part of what we've done on also on the platform. This is the cell dive process, as David showed you just before. I won't go into details about it because of David, what I told you. But just to show you one part is that the slide preparation, we've included our automated stainer to do the baking, the clearing, antigen retrieval, the blocking, and the DAPI staining. Of course, once this part is done, we can include also this device for the biomarker labeling and the dye inactivation during all the runs, different runs. And this reduces time and ensures standardized labeling. Thank you.
Thank you so much, Nicola and David. Those were excellent presentations. We've got a few questions from the audience. If anyone else has a question, you can leave those in the questions box that appears on your screen. So the first question to start you off, and you can choose who wants to answer this. I think this might be one for Nicola. Which kind of fluorophore can be used with the cell dive?
You've got 4 fluorophore. You've got the cyanine 2, the cyanine 3, 5, and 7. But if you don't have the cyanine, then except for the cyanine 2, then you can use hexafluor, for example, and many others. Perhaps David want to add informations?
Yeah, thanks. That's a lot of different fluorophores, at least. The next question is, what is the amount of data that's generated by one acquisition?
Oh, it depends on the how many proteins you want to look at. For example, what I've just showed you before with the 9 markers, we were about 80 gigabytes of data.
Okay. Yeah, and I guess, yeah, it makes sense that it depends on what you're actually looking at.
Yeah, of course.
These are a few questions that came in during David's talk, so he might be best placed to answer them. The first one is, can the repeated process of staining and inactivation be a problem for TMAs where spots are more fragile than standard tissue sections?
So it won't be a problem. It has been part of the validation. It has been validated on, yeah. So FFP mutation, either the whole tissue or the TMA. We have a lot of users that that use successfully the TMA. It's a great also to scale up this kind of hyperplexing technique. So it's definitely validated for TMAs.
Okay, great. Thank you. And does CellDive work with frozen sections?
So all the process has been validated with a FFPUT section, but we have some users that started to investigate the use of the fresh frozen section with the CellDive process. So it's on progress, in fact.
Okay, thank you. There are a few questions here, so I'm just reading through them to make sure I'm asking them all separately. Do I have to buy pre-labeled antibodies for this?
So antibodies, it's a trend also of the biomarkers that are available in the market. They are more and more available directly conjugated, which is great. Otherwise, you will have to make the conjugation by yourself. And this is also something that we document in the documentation of the CellDive. It's not rocket science, it's easy to conjugate. And you just have to follow the process.
Okay, thanks. And is it possible to export images to analyze them in other software?
So for now, as we partnership with Indica Labs, the output is directly compatible with the Halo software. So that means that the system generates an XML file and you can just open the XML file into Halo. On the other side, we are also working on the capability to define the output that you want to fit best the imaging solution that you have on site.
Thank you. Nicola, let's go back to you for a question. What objectives are available on the scanner?
You've got 3 different objectives. The 2x, the 10x, and the 20x. And yeah, that's it.
Okay, so 3 sizes. And is it a confocal?
No. Okay. It's not a confocal. We've got a confocal scanner on the platform, but the CellDive is not a confocal scanner.
And I guess you don't need it for these types of analysis. I've got a number of questions that came in all at once at the end of the presentation. So let me just go through some of these. If a biomarker is not abundant, is it possible to visualize it using CellDive tech?
It will be possible to detect a biomarker which is not abundant. Mainly because actually we characterize the autofluorescence that can really compromise the dim signal from the biomarkers. So thanks to the precise characterization of the autofluorescence, we will be able to detect dim biomarkers. Also thanks to the microscope platform that we have developed, which is super sensitive, but I think it's a combination between the microscope itself and the method.
Okay. And that actually ties into one of the other questions. How does the sensitivity compare with other amplification-based systems?
So we don't use yet any amplification method. We use primary antibodies directly conjugated with fluorescent dyes. And again, thanks to the autofluorescence characterization and also the advanced calibration, it's a way to get very bright and very precise images coming from this different biomarker. It's a super sensitive system. It's a super sensitive wide field microscope as well.
That's great. And how many slides can you do at the same time? So how long does it approximately take to do one experiment?
So actually, the good thing is that you can really separate the different cycles. I mean, during that you will take care of the staining or the dye inactivation of a given slide, you can image another slide. We selected that to make always available the imager to take images. And thanks to that, you can interleave different slides and different projects. So in one day, you can do a multiple cycle, but also from different, coming from different slides.
Great.
And Nicolas mentioned also the total number of days that he took to conduct the analysis that he presented to us.
Yeah, thank you. And then we've had a few questions about the validated antibodies. How can we find the list of the validated mouse antibodies and how many validated antibodies are available?
So the validated antibodies, they really come with the cell dive, with this workflow solution. It's they are all together with the imager, the workflow, and the validated list. Until now, we validated 350 different biomarkers, and I would say about 90% for human, because this is all our expertise and history that we have built, and we have 10% of mouse antibodies. And as Nicolas said, it's also easy and actually open to add more antibodies from yourself, from your lab.
Great. I've got a few questions left. So let me have a look at this. How can you be really sure that fluorescence is inactivated?
So it's part of the validation. That means that once an antibody has been validated, we also validated the fact that the fluorescence can be inactivated. And also because we image the slide after the dye inactivation, it's also a way to test on the current sample how the inactivation went.
Great, thank you. And then our final question for today is, you mentioned that you can acquire the entire tissue section on a slide that's very big. And how do you deal with the focus for this large area?
So for that, we have developed a specific focusing method that uses both what we call hardware autofocus, where a laser light will actually detect the interface between the glass slide and the tissue, combined with a software autofocus, because you never know if the tissue will be perfectly attached to the glass. So this is what we will do actually for every field of use. And before to take that, it was also shown on my video, there is a tilt check that will actually characterize the focus map of the slide by exploring different points to already have like a focus map prediction.
Yeah, that's a clever idea, thanks. And that brings us to the end of our seminar today. So thank you again, David and Nicolas, for a very illuminating presentation and a great discussion afterwards. And thank you to our sponsor, Leica Microsystems. And finally, of course, thanks to you, the audience, for taking the time to attend and listen in today. So until next time, good luck in your research and goodbye from all of us at Leica Microsystems and Bite Size Bio.